and this is the experimental framework of this study. So there were 3 skin-related cells were cultured in this experiment. They are the human keratinocyte cell, human dermatofibroblast cell, and the B60F murine melanoma cell, respectively. And the HACA cell and HGF cell were subjected with the UV irradiation and treated with the tendinized Lactobacillus acidophilus in the dose-dependent manner. And the B60FM murine melanoma cell was stimulated by the alpha MSH and then treated with the tendinized Lactobacillus acidophilus KCL12625P in the independent dose manner. And the cell-availability assay was to determine the cytotoxicity of the AL to the skin-related cell, and to investigate whether the AL reduces the RS generation. The H2DCF-DS staining assay was used in this experiment, and the antioxidant effect of AL was further investigated in the white spot skin by using FDS radical scattering assay. And skin-related gene expression conducted the RT-PCR to determine the anti-wrinkle effect and the expression level of the anti-wrinkle related genes in this study. And the Western blotting also was conducted to determine the antioxidant effect of the AL. And ELISA was used in this experiment to confirm the expression of matrix metaproteinases, and elastase in vitro assay was conducted to determine the effect of AL to exert its anti-wrinkle effect on the skin. And the anti-melanogenesis effect was determined by the melanin generation assay, and tyrosine is the one of the important molecule in the production of melanin, so the tyrosine activity assay was used. And this is the mechanism of the skin wrinkle formation. So repeated exposure to UV irradiation will increases the generation of RS, and RS will induce the phosphorylation of P38 and ERK through the activation of an MPK pathway. And the activation of P38 and ERK will induce the phosphorylation of C-Jun and C-Phos through the active signaling pathway, and induce the expression level of the matrix metaproteinases 1 and cause the degradation of collagen and breakdown and cause the formation of wrinkles. And this is the mechanism of melanogenesis. So when the skin repeated exposure to UV radiation will cause the DNA damage in the keratinocyte and induce the P53 to stimulate the P53 protein to induce the expression of POMC, which is the precursor protein of the alpha MSH. And alpha MSH will interact with the MC1R to induce the reaction of CMP signaling pathway in melanocyte in order of the protein kinases and CMP responses element binding and ITF to increase the expression of tyrosinase to induce the production of pigmentation such as melanin and melanocyte. So this is the result and discussion. So in figure 1 is to show the antioxidant effect of tendinized Lactobacillus acidophilus as an aberration of AL in the white spot skin. So figure 1A display the human keratinocyte cell were subjected by the UV irradiation in 30 mg per cm squared, and in the absence of presence of AL in the concentration range from 50 to 200 microgram per liter. And the result was determined by H2DCF-DS staining assay. And the result shows that the R generation of the HACA cell was firstly induced by the UV irradiation and then recovered by the AL in the dose-dependent manner. And in figure 1B shows the cell availability of HACA cell was firstly treated by the indicated dose of AL in the concentration range from 25 to 100 microgram per milliliter, and the result was measured using the MT assay. And the MT assay shows that the AL had no any cytotoxicity to the HACA cell in 24 hours. And in figure 1C display the HACA cell were firstly subjected by UV irradiation and then treated in the indicated dose of AL for 24 hours and also determined by the MT assay. And the result confirmed that, and the result shows that the cell viability of HACA cell was firstly decreased by UV irradiation and then recovered by AL in dose-dependent manner, which implies a cytoprotective against cell death caused by the oxidative stress. And in figure 1D display the FDS radical scattering activity of AL in the indicated dose of ATDS from 25 to 400 microgram per liter, and Haga cell was incubated with ATDS with either AL or ascorbic acid as a causative control. And the result shows that the AL reduced ATDS related activity in a density dependent manner. So taken together, this data strongly suggests that the AL had an antioxidant effect. And in figure 2a shows the following pre-treatment of AL of the elastase inhibition activity on the Haga cell was incubated with elastase and SANA and urusolic acid as a causative control. And the result shows that the AL inhibited the elastase activity in the dose-dependent manner. And in figure 1b described that the Haga cell was firstly subjected by UV irradiation and then treated in the indicated dose of AL in the concentration range from 50 to 200 microgram per milliliter, and the expression level of AMP1 was determined by ELISA, and the result shows that the expression level of AMP1 was firstly induced by UV radiation and then inhibited by AL in the dose-dependent manner. And in figure 1c shows the mRNA expression of the matrix member proteinases 1, 2, 3, 9, and SERT1 with subjected by UV irradiation and then treated in the indicated dose of AL for 24 hours and taken using the PCR analysis. So the result shows that the AL inhibited the expression level of the matrix member proteinases 1, 2, 3, and the expression level of SERT1, which has a notable function of cell survival and inflammatory processes, as increases approximately to its baseline level. And in figure 2d shows the Haga cell was treated, the ER head and C-FOS protein was in the Haga cell was subjected by UV irradiation and then treated in the indicated dose of AL from 50 to 200 microgram per liter and was determined by the Western body, and the result shows that the phosphorylation of ERK and C-FOS was significantly inhibited by AL in the dose-dependent manner. So in figure 3a shows a human dermal fibroblast cell was treated by AL in the concentration range from 25 to 400 micrograms per liter. And the result was determined by the MT assay for 24 hours, and the result shows that the AL had no any cytosity to the HDF cell. And also in figure 3b shows the HDF cell was treated by the AL in the concentration range from 25 to 400 micrograms per milliliter for 48 hours. And the result was determined by the MT assay. And the MT assay shows the cell availability of the HDF cell either suppressed by AL in the concentration used. So it shows that the AL had no any cytosity to the HDF cell during these 48 hours. And in figure 3c display the elastic inhibition activity. So in the following pretreatment of AL in the concentration range from 50 to 200 micrograms per liter, and were subjected by UV irradiation in 30 mg per cm squared, and the result shows that the AL inhibited the elastase activity in the dose-dependent manner. And in figure 3d display the expression level of matrix member proteinases 1 was firstly subjected by UV irradiation in 30 mg per cm squared and then treated in the indicated dose of AL in the concentration range from 50 to 200 micrograms per liter, and was evaluated by using ELISA, and the result shows that the expression level of AMP1 was firstly induced by UV irradiation and then suppressed by AL in the indicated dose manner. And in figure 3E and 3F display the Taiwan propagation alpha was treated by AL with either the subjected by UV irradiation for 30 mg per cm squared in 24 hours. And the result shows that the AL increases the expression level of Taiwan propagation alpha in figure 3E, and in figure 3F shows the result of Taiwan propagation alpha expression level was firstly decreased by UV irradiation and then recovered by AL in the indicated dose manner. And in figure 3G shows the mRNA expression of the matrix member proteins 1, 9, and COL1A1 with subjected by UV irradiation and then treated by indicated dose of AL in the concentration range from 50 to 200 microgram per milliliter for 24 hours. And the result was determined by the RT-PCR. And the result shows that the matrix metaproteinases 1 and 9 was significantly inhibited by the AL in the concentration used, and the COL1A1 expression level was significantly increased by the AL in the dose-dependent manner. And in figure 4a, display the B60F10 murine melanoma cell was treated by the AL in the concentration from 50 to 200 microgram per milliliter for 48 hours, and also the result was determined by using the tetrazolium calorimetric as the average of anti-acidic, and the result shows that the AL didn't show any cytotoxicity to the B60F10 murine melanoma cell in the 48 hours. And figure B shows the B60F10 murine melanoma cell was firstly stimulated by the alpha MSH, the alpha melanocyte stimulating hormone, and then treated by AL in the independent dose concentration range from 50 to 200 microgram per milliliter, or either arbutin as a positive control, and the result was measured by 475 nm. And the result shows that the melanin secretion in the B60F10 murine melanoma cell was decreased by the AL in the independent dose lag. And in figure 4c, display the intracellular melanin content of the B60F10 murine melanoma cell was firstly stimulated by alpha MSH and then treated in the indicated dose of AL in the concentration range from 50 to 200 microgram per milliliter, or either treated by arbutin as a positive control. And the result shows that the intracellular melanin content of B60F10 murine melanoma cell was firstly increased by stimulated by the alpha MSH and then treated by recovered by AL in the dose-dependent manner. And in figure 4D shows the mushroom tyrosinase was incubated with the L-DOPA and treated with the folic acid as a positive control, or either AL in concentration range from 50 to 200 microgram per milliliter. And the result shows that AL had no any inhibition due to the tyrosinase activity. And in figure 4E, display the tyrosinase protein 1 and tyrosinase protein 2 from the B16F10 murine melanoma cell was firstly stimulated by alpha MSH and treated in the independent dose of AL in the concentration range from 50 to 200 microgram per milliliter. And the result was determined by the RT-PCR analysis. And the result shows that the expression level of tyrosinase and tyrosinase protein 1 was significantly inhibited by AL, but not the tyrosinase protein 2. So this data indicate that the AL did not inhibit the tyrosinase activity directly. And in figure 4F, display the protein expression level of the various cyclic adenosine monophosphate signaling pathway, for this in the response to the AL in the concentration range from 50 to 200 microgram per milliliter. And the result was determined by using the Western blotting. And the result shows that the phosphorylation of PKS, CREB, MITF, and tyrosinase was inhibited by AL in the dose-dependent manner. And this is the minor article from figure 1B and C. So display the B60F10 murine melanoma cell was treated by alpha MSH, and the melanin secretion and melanin content was measured by using isomers of 475 and 405 nm. And the result shows that the melanin secretion and melanin content was suppressed by the Hannah-Stimson-Calyx agonal extract in the dose-dependent manner. And conclusion. So this is the mechanism of the anti-radical anti-melanogenesis effect of individualized Lactobacillus acidophilus, and the arrow indicates positive regulation and T-bar indicates negative regulation. So according to administration of AL impeded the R strain production by using the R generation assay. And this study showed that the antioxidant effect of AL for the skin and body from the UV radiation by using the NTS radiation scattering assay. And the cell array assay demonstrate that AL did not show any cytosity in the concentration used for the cell and could also prevent the cell from the death by UV oxidative stress. And the RT-PCR analysis was conducted and demonstrate that AL increased the mRNA expression of SIRT1, which has the notable function in the cell survival and inflammatory processes. And the ELISA was already demonstrated that Taiwan population alpha level increased by AL, which has the function of modulate the skin tissue structure. And the postmortem body demonstrated that the AL regulated the protein level of CMP signaling pathway to exert its anti-melanogenesis effect. And this is the conversion between the main article and minor articles. So the concentration use of AL loss from the main article was lower than the concentration use of the skin cell external extract.